imagine two human patients, okay?
Right.
Both of them have just been diagnosed with diffuse pleural mesothelioma, which is, a really aggressive, highly lethal cancer of the lung lining. It's the one usually associated with asbestos exposure.
Yeah, a devastating diagnosis.
Truly.
Yeah.
So both of these patients are given our absolute best, state-of-the-art weapon right now, anti-PD-1 immunotherapy. And the whole clinical goal here is to basically take the brakes off their own immune systems, right? So their CD8 killer T cells can just flood into the area and eradicate the tumor.
That's the ideal scenario, yes.
Right. And for patient A, the therapy works beautifully. Their tumor is just swarming with killer T cells and it actually starts to shrink.
Yeah.
But then, and here's the tragedy for patient B, the therapy completely fails.
It just doesn't do a thing.
Exactly. The cancer progresses. And if you take a biopsy of patient B's tumor and look at it under a microscope, there are almost zero killer T cells in there. Instead, the tumor is absolutely packed with this specific type of macrophage. We'll get into the details, but it's a CD163 positive macrophage that basically acts as a giant off switch for the immune system.
It's a localized immunosuppressive zone.
Yeah. And so today, we are doing a deep dive into a really groundbreaking 2026 study published in the journal Med by Lopez Lago and colleagues, because they figured out why this happens. They looked at a stack of data and revealed that the difference between life and death for patient A and patient B, the reason one responds and the other doesn't, comes down to a single missing gene.
It is a remarkable piece of research because it completely upends, our traditional view of therapy resistance.
How so?
Well, for decades, when a tumor resisted a drug, we kind of just assumed the cancer cell itself had mutated to build a stronger armor, right? Maybe it changed its surface protein so the drug couldn't bind, or it developed a way to pump the chemotherapy back out of the cell.
Like a soldier putting on thicker Kevlar.
Exactly. A cell autonomous defense. But this data shows a completely different, almost sinister strategy. The mutation we're talking about today doesn't change the cancer cell's personal armor at all. Instead, it gives the cancer the ability to radically brainwash the surrounding healthy tissue.
Okay, wow. So we were talking about a non-cell autonomous resistance mechanism.
Right.
The cancer isn't protecting itself. It's manipulating the local environment, its neighbors, to do the protecting for it. And the culprit behind this, the genetic trigger that starts this whole cascade, is the loss of a gene called NF2.
That is the core finding, yeah. So NF2 is a classic tumor suppressor gene, and we've actually known for a long time that it's frequently mutated or just completely lost in mesothelioma. Now, NF2 codes for a scaffolding protein called myelin. And you can sort of think of myelin as this very strict cellular supervisor. Its job is to keep cell division and internal signaling pathways in check.
Like the foreman on a construction site.
Precisely. So when a cell loses the NF2 gene, that supervisor is just gone, and the internal wiring of the cell goes completely haywire. But what Lopez Lago's team discovered is that this internal chaos doesn't just make the cancer cell grow faster, which is what we used to think. It actually turns the cell into a masterful manipulator of the immune system.
Let's trace the path of that discovery, because the methodology here is just fascinating. They started by intentionally silencing the NF2 gene in human mesothelioma cells, right? Just to see how they would react to TRT cells.
Right. They used a technique called shRNA to deplete it.
And if I'm a researcher and my running hypothesis is that losing NF2 causes therapy resistance, my first instinct is that these NF2 depleted cancer cells will suddenly become bulletproof in a petri dish.
Oh, absolutely. That's the logical assumption. And it is exactly what they did not see. Yeah. So they ran a standard chromium-51 cytotoxicity assay, which is basically just putting the NF2 deficient cancer cells and the killer RT cells together in an isolated lab dish. And the CRT cells just absolutely slaughtered the cancer cells without any issue at all.
Wow.
Yeah, losing NF2 did absolutely nothing to protect the tumor cells in a vacuum. The cancer cells were just as vulnerable as ever.
Which is the massive aha moment of the paper.
Right.
Because in isolation in the petri dish, the cancer is weak, but in the human body, in patient B, the tumor survives.
Exactly.
So the researchers knew they had to move into living models to see what was actually happening in the biological neighborhood, what we call the tumor immune microenvironment, or the TIME.
The TIME, yes. It's a very fitting acronym.
So they took these NF2 depleted mesothelioma cells and injected them orthotopically into the pleural cavity of mice, meaning they put them in their natural anatomical home, the lung lining.
And just to be thorough, they actually used 2 distinct mouse models to ensure their findings weren't just a fluke. They used immunodeficient mice, NSG mice, injected with human cancer cells, but they also used an immunocompetent syngeneic model.
Wait, what does syngeneic mean for those listening?
Right. It means mice with fully intact, healthy immune systems injected with mouse mesothelioma cells. So it's a completely natural immune interaction. They even engineered the cells with firefly luciferase so they would emit light.
Well, that's cool. So they could literally watch the tumor grow in real time under imaging.
Exactly. And the results in vivo in the living mice were just night and day compared to the petri dish. When they administered CAR-T cells or anti-PD-1 therapy, the tumors that still had an intact NF2 gene shrank. The therapy worked, but the tumors missing NF2, they relapsed rapidly. The mice succumbed to the disease much, much sooner.
Okay, so if I'm understanding the biology here, the tumor cell is not the soldier in Kevlar. It's more like a mob boss.
A mob boss, I like that.
Yeah. Think about it. The boss himself is totally vulnerable if you catch him alone in an alley, which is basically what happened in the petri dish, right?
Right, isolated.
But in the real world, the boss never walks alone. He hires local bodyguards to surround him and intercept any threats before they even get close.
That is a very apt way to visualize non-cell autonomous defense. The tumor is literally exploiting its neighbors, and the researchers actually went in and identified exactly who those bodyguards are.
Who are they?
So when they harvested those resistant tumors from the pleural cavities of the mice and analyzed the cellular makeup, they found a massive influx of macrophages. Specifically, these were marked by F480, which is just a standard macrophage marker in mice. But they also heavily expressed a receptor called CRIG.
CRIG? Let me make sure I have this right. That stands for complement receptor of immunoglobulin superfamily.
You got it. And the presence of CRIG is a huge red flag. It indicates a highly, highly immunosuppressive subtype of macrophage. These are the cells that physically crowd around the tumor and actively shut down the killer T cells. So somehow, the loss of this one gene, NF2, inside the tumor cell triggers this massive recruitment of SRIG-positive macrophages into the neighborhood.
Okay, but I have to push back on this whole mob boss controlling the neighborhood idea for a second.
Go for it.
Because if a tumor has this incredible, almost magical genetic ability to summon an army of suppressive bodyguards just by losing one gene, does it have this power everywhere? Like, if a metastatic mesothelioma cell lands in the liver or the bone marrow, can it pull the exact same trick?
Yeah, that is perhaps the most profound question the researchers asked. And the answer completely invalidates a lot of assumptions we make in oncology today.
Oh, really?
Yes. They took those exact same NF2 deficient mesothelioma cells, the ones that easily defeated CAR-T cells in the lung pleura, and injected them subcutaneously, just under the skin of the mice.
They changed the anatomical zip code.
They moved the tumor to a different neighborhood. And the resistance mechanism completely evaporated.
Wait, really?
Yes. Under the skin, the CAR-T therapy worked flawlessly. The tumors did not relapse. And when they analyzed the tissue surrounding those subcutaneous tumors, there was no massive influx of SRIG-positive macrophages.
That is staggering. The tumor's ability to evade the immune system is entirely dependent on its physical location. It's an anatomical site-specific resistance.
Exactly.
So the tumor can only hire those specific bodyguards if it's sitting in the unique immunological environment of the pleural cavity.
Which highlights a major flaw in how we historically test cancer drugs. For decades, researchers have relied on subcutaneous xenograft models, right? You inject a tumor under the skin of a mouse because it's easy to see and measure with calipers.
Right, it's convenient.
But this study proves that a drug might look incredibly successful against a subcutaneous tumor in the lab, only to fail completely in a human when that same tumor is growing in its natural organ, surrounded by a completely different cast of local immune cells.
Okay, so let's pause here for a quick recap because we cover a lot of ground quickly.
Good idea.
So bullet point summary for you listening at home. First, NF2 is a tumor suppressor gene, and losing it in pleural mesothelioma causes resistance to CAR-T cells. Second, it does this not by changing the cancer cell's direct armor, but by remodeling the local neighborhood the time to recruit these suppressive CRIG-positive macrophages.
The bodyguards.
The bodyguards. And third, this trick only works in the lung pleura, not under the skin. It is site-specific. So here is a question for you to mull over. If you were designing a cancer drug, would you focus on attacking the tumor cell itself, or would you try to reprogram the neighborhood it lives in? Because it seems like the neighborhood is where the battle is actually won or lost.
It really is.
Okay, moving into the second phase of this deep dive, we've established that the loss of NF2 turns the tumor into this master manipulator, but only on its home turf. The obvious next question is how?
The mechanism of communication.
Right. What is the actual chemical text message? Because the tumor cell doesn't have vocal cords. It has to be sending some kind of physical signal into the environment to summon these bodyguards.
Well, to decode that signal, the team had to look at what happens inside the cancer cell the exact moment the NF2 supervisor goes missing. And without that myelin protein, two major intracellular signaling pathways spiral completely out of control.
Okay, what are they?
The first is the Hippo pathway, specifically a transcriptional complex called YAPTID. The second is the MTOR pathway, which regulates cell growth and metabolism.
And we usually measure MTOR hyperactivity by looking at a downstream target called PS6, right?
Correct. So you have YAPTID moving into the nucleus to wildly alter gene expression, and MTOR driving these aggressive metabolic changes. This internal dysregulation basically forces the tumor cell into a state of panic, causing it to pump out specific chemical messengers into the surrounding tissue.
It's essentially hijacking the local emergency broadcast system. First, it sends out an SOS signal to lure the first responders in. And the researchers actually found out exactly what that SOS signal is through RNA sequencing.
They did.
They found that NF2 deficient tumors secrete massive amounts of myeloid-attracting chemokines. In mice, it's a chemokine called CCL9. And in human cells, the equivalent signal is CXCL8.
Those chemokines are incredibly powerful attractants. They diffuse out into the surrounding pleural tissue and physically pull circulating macrophages toward the tumor site.
Right.
But here's the thing. Simply pulling the macrophages in isn't enough to build an invisible shield, because a normal, healthy macrophage might arrive, recognize the tumor as a threat, and try to destroy it.
Yeah, the first responders show up expecting to put out a fire. The tumor has to somehow brainwash them into becoming the mob boss's bodyguards.
And that brainwashing requires a second, distinct chemical signal. The researchers discovered that the loss of NF2 also radically upregulates an enzyme inside the tumor cell called ALDH1A3.
ALDH1A3.
Yes, and this enzyme has a very specific metabolic function. It synthesizes retinoic acid.
Retinoic acid. Like the same molecule we hear about in skincare and dermatology for cell turnover?
The very same. But here, in the tumor microenvironment, it's acting as a potent local hormone, a polarizing signal.
Okay, explain that.
When those recruited macrophages arrive at the tumor site, they're suddenly bathed in high concentrations of retinoic acid. This forces them to differentiate. In immunology, we generally classify macrophages into two broad states. M1, which are pro-inflammatory anti-tumor cells that help killer T cells do their job.
The good guy.
Exactly. And M2, which are immunosuppressive wound-healing cells that shut down immune responses to prevent tissue damage.
And the retinoic acid forces them into that M2 state.
It forces a severe M2 polarization, causing them to express that CI receptor we talked about earlier. So the tumor uses the chemokine CXCL8 to pull the macrophages in, and then uses retinoic acid to fundamentally alter their identity, turning them into a suppressive wall that CRT cells cannot penetrate.
Let me try to synthesize this with a visual mnemonic, because the mechanism is brilliantly sinister, and there are a lot of acronyms floating around.
Let's hear it.
Okay, let's move away from the mob boss. Picture a pirate ship. The pirate ship is our NF2 deficient tumor. The Hippo pathway is the broken steering wheel, causing the ship to spin out of control.
Okay.
So the ship drops a chemical anchor, that's the CCL9 or CXCL8 chemokine, to stop passing macrophages. And then once they're close, it fires a retinoic acid flare produced by the ALDH1A3 cannon to basically hypnotize the passing ships into becoming loyal M2 bodyguards.
That is a surprisingly accurate analogy. That is the complete non-cell autonomous mechanism.
But in science, proposing a mechanism based on molecular observation is really only half the battle. The burden of proof requires demonstrating causality. You have to prove that intercepting these specific signals actually breaks the shield.
Because if you see a fire engine at a fire, it doesn't mean the fire engine started the blaze.
Exactly. So how did the team definitively prove that the chemokines and the retinoic acid were the actual cause of the resistance, and not just biological bystanders?
They systematically dismantled the tumor's communication network using both genetic engineering and pharmacology. First, they tackled the SOS signal. They engineered a line of double knockdown mesothelioma cells.
Double knockdown?
Yeah, they removed the NF2 gene, so the tumor wanted to build a shield, but they also deleted the gene responsible for the CCL9 chemokine.
They cut the radio tower's transmission wire.
Exactly. And when they injected these double knockdown cells into the pleural cavity of mice, the results were definitive. Without the chemokine signal, the macrophages never arrived. The local environment remained unprotected, and the SRR T cells were able to flood in and clear the tumor. The mice survived.
So that proves the first step, the chemical anchor. But what about the brainwashing step? What if the macrophages were already there, but you stopped the retinoic acid flare?
To test that, they turned to a pharmacological approach. They used a compound called DEAB, which is a specific inhibitor of that ALDH1A3 enzyme. DEAB essentially jams the machinery that produces retinoic acid. They took mice with fully established NF2 deficient tumors that were actively resisting therapy and treated them with DEAB alongside the CAR-RT cells.
And the flow cytometry data here is just beautiful. By blocking the retinoic acid, the macrophages that had gathered around the tumor stopped polarizing into that suppressive M2 state. They never expressed CRIG.
Right, the brainwashing stopped.
Without that suppressive brainwashing, the microenvironment flipped from hostile to permissive, and the CAR-RT cells regained their lethal function.
It is a stunning validation of the pathway. It proves that therapy resistance in this context is an active, ongoing process of chemical communication. If you disrupt the communication, you strip the tumor of its defenses.
Which is incredible. So let's pause for our second recap.
Sounds scary.
Bullet points. NF2 loss dysregulates the Hippo and mTOR pathways inside the cell. This broken steering wheel forces the tumor to pump out chemokines, like CXCL8, to pull in macrophages, and retinoic acid, via ALDH1A3, to polarize them into M2 bodyguards. Blocking these signals with gene editing or drugs like DEAB completely strips the tumor of its shield.
Perfect summary.
And here's the question for you to think about this time. Can you think of other systems, whether in biology or even in society, where intercepting a distress signal is actually a more effective strategy than directly attacking the source of the problem?
It's a very systemic way to view conflict.
Yeah. All right, moving to the final subtheme. This is the most critical leap in any preclinical study, moving from mice to human beings.
The translational hurdle.
Because everything we've discussed so far relies heavily on engineered cell lines and mouse models. But the immune system of a mouse is notoriously different from ours. Does this highly localized, complex brainwashing actually happen in human lung tissue?
To answer that, the researchers had to look at massive amounts of human genomic data. They utilized a highly sophisticated computational method called CyberSort. CyberSort stands for cell type identification by estimating relative subsets of RNA transcripts.
I love the concept behind CyberSort. I always explain it like it's a mathematical algorithm that lets you unbake a cake.
Unbake a cake.
Yeah. You take a solid block of bulk tumor tissue, which is like a baked cake, sequence all the RNA inside it, and the algorithm calculates exactly how many immune cells and what specific types of immune cells, the flour, the eggs, and the sugar, are mixed into that tumor based on their unique genetic signatures.
That is actually a very accurate way to describe transcriptomic deconvolution. I might steal that. So they applied the CyberSort algorithm to two massive human databases, the Cancer Genome Atlas and a specific cohort of mesothelioma patients from Brigham and Women's Hospital.
A huge data set.
In total, they analyzed the tumor microenvironments of 284 human patients.
And the human data perfectly mirrored the mouse models.
Right. It was incredibly consistent. They found that in the 66% of human patients who had an NF2 copy number loss, meaning their tumors were missing the supervisor gene, the CyberSort data revealed a statistically massive abundance of those M2 tumor-associated macrophages.
The bodyguards were there.
The bodyguards were there in force. And at the same time, there was a drastic reduction in the presence of CD8-positive killer T cells. The human plural neighborhood was being corrupted exactly as the mouse models predicted.
But they didn't stop at just identifying the cells. They took it a step further to see how this actually impacts human survival. They built an mRNA-based anti-PD-1 resistance signature.
Right.
They basically looked at the gene expression of human patients who had received immunotherapy in the clinic. They separated the patients whose tumors shrank from the patients whose tumors aggressively progressed, our patient A and patient B, from the very beginning of the show.
And when they mapped that clinical response data back against the genetic profiles, the correlation was undeniable. Human tumors with an NF2 loss are fundamentally, structurally wired to resist anti-PD-1 drugs because their microenvironment is preemptively flooded with suppressive macrophages.
This is where the science hits the clinic. Because we can't just run a genetic test on a patient, see they're missing NF2, and say, well, sorry, standard immunotherapy won't work for you. We have to use this granular understanding of the mechanism to reverse engineer a cure. So what is the ultimate therapeutic strategy here?
Based on the mechanisms they uncovered, the study proposes 3 distinct actionable points of vulnerability to break the shield and restore the power of immunotherapies in NF2 deficient tumors. We call them the 3 pillars of reversal.
Okay, 3 ways to cut the communication. What's the first target?
The first target is upstream, inhibiting the YAP-TED complex. Remember, when NF2 is lost, the Hippo pathway crashes and YAP-TED goes into overdrive, setting off the chain reaction.
The broken steering wheel.
Right. And there are actually TED inhibitors currently in early phase clinical trials for other solid tumors. The data here suggests that giving a patient a TED inhibitor wouldn't necessarily kill the tumor directly, but it would stop the initial panic response, preventing the tumor from ever remodeling the microenvironment.
You fix the steering wheel before the SOS is sent. What is the second pillar?
The second is intercepting the SOS signal itself through CXC blockade. If we use targeted drugs or neutralizing antibodies to block CXCL8 in humans, the chemokines can't pull the macrophages toward the tumor site. The bodyguards never get the text message to show up.
Okay, and the third target?
The third is targeting the brainwashing phase using retinoic acid inhibition. If we use ALDH inhibitors similar to the DEAB compound used in the lab, we can stop the tumor from synthesizing retinoic acid.
Putting out the flare.
Exactly. Even if the macrophages arrive at the tumor without the retinoic acid, they remain neutral, or they might even adopt an M1 state and help attack the cancer.
So the future of treating this highly lethal cancer isn't really about inventing a single magical drug. It's about combination therapy. It's about precision oncology.
It absolutely is.
We use CAR T cells or anti-PD-1 drugs as the weapon, but we have to pair them with a TED inhibitor, a chemokine blocker, or an ALDH inhibitor to strip away the tumor's defensive ecosystem first.
That is the paradigm shift. We have to treat the microenvironment as actively as we treat the malignancy itself, and it completely changes how we should be thinking about clinical trial design. If we know a patient has an NF2 mutation, we shouldn't waste precious time giving them monotherapy immunotherapy that is biologically destined to fail.
Right, you can preemptively disarm the tumor's defenses from day one.
Exactly.
Okay, final recap before we wrap up.
Let's do it.
Bullet points. Human genomic data checked via the CyberSort algorithm across 284 patients completely confirms the mouse models. NF2 loss means more M2 macrophages, fewer killer T cells, and failed immunotherapy. The future of treatment lies in combining our current weapons with drugs that block TED, chemokines, or retinoic acid.
A very clean summary.
And your final question for this section. Knowing this, how might a routine genetic test for NF2 status completely change the trajectory of a patient's personalized treatment plan from the very moment they are diagnosed?
It gives them a fighting chance.
It's an incredible evolution in how we view illness. We started this deep dive looking at the frustrating reality of a failing treatment. But studies like this are giving us the high-resolution X-ray vision. We need to see exactly where the biological systems are breaking down and exactly which wires we need to cut to fix them.
It's empowering.
It is. It is no longer just about engineering a sharper weapon to stab a cancer cell. It's about understanding that the tumor is actively engaging in psychological warfare on a cellular level. You have to win the neighborhood if you want to win the war.
And that concept of the neighborhood, the anatomical constraints of disease, is perhaps the most vital takeaway for the broader field of medicine.
Absolutely. And that is the expansive thought we want to leave you with today. Think back to that incredible detail about site specificity we talked about earlier.
The subcutaneous versus orthotopic models.
Yeah. The tumor's ability to broadcast its SOS, pull in macrophages, and brainwash them with retinoic acid. It only worked in the lung pleura. When the exact same tumor was moved just an inch away, under the skin, the shield completely failed. The local zip code dictated the success or failure of the immune evasion.
The tissue specificities of our immune system are vast and largely uncharted.
They really are. So if a tumor can manipulate immune cells based entirely on its anatomical zip code, what other local tissue environments across our bodies are silently dictating the success or failure of our daily medications, our diets, or our standard therapies without us even realizing it?
It's a huge question.
How much of medicine have we misunderstood simply because we were looking at the right cell in the wrong neighborhood? It's a massive frontier for us to explore. Thank you for joining us on this deep dive.